Thank you very much for the invitation, Philip, and the organizers. I've often asked to talk about in conferences about personalized medicine, and the focus is often on late cancers, advanced disease, sequencing the mutations, and matching patients to drugs. But more recently, I decided there's lots of other aspects of large-scale genomics, which actually probably informing us about the early cancers, which is also probably important for prevention and early detection. And I decided to focus my talk on those topics. So you're going to ask me for a definition of early cancers. Well, I'm going to just say if it's still localized to the original tissue, I'm going to call it early. But we know actually that, it's not late, not outside the original tissue, but the early cancers actually is a process that takes many years. Actually, we can inherit mutations which are driver mutations, or we can acquire them during our lifetime. And if we smoke or have certain exposures, we can even accelerate the process of getting mutations. Mutations is certainly the hallmark of cancer. And I'll give examples. I really wanted to give examples of once you have a big data sets, how you can get to some biology, which is new, and also some of the clinical information, which also might be informative for the clinician and the patient. When we do talk about cancer, we talk about two genomes. In fact, we talk a lot more than two genomes because every cancer cell seems to have some differences between each other. But certainly we talk about the germline genome, the inherited genome from parents, from an individual has, but also the genome of the tumor, which has many changes. So I'm going to start talking about germline variation, genetics actually predisposed to colon cancer. Then I'm going to talk about somatic mutation examples. So we've been doing GWAS now for about a decade, since the HapMap project, and there's thousands of loci have been identified. And actually, there's a lot of commonalities when we associate a polymorphism. Really is a tag of a haplotype or region of linkage equilibrium with the phenotype. And most of what's been discovered actually has very low risk compared to the germline mutations, which actually gave rise to cystic fibrosis and many of the diseases that Guy studied many years ago, or still studies today, the pediatric disease having very penetrant alleles, which affect protein structure. Most of the GWAS alleles actually have very low effect, increasing risk 10 or 20%. Most of the loci have been identified of the 2,000 for common diseases and 400 or 500 for cancer. Actually, we still have not identified exactly what is the gene, the causal gene. That's going to be a bit of my topic for today. And sometimes we've seen genes we've seen before, but most of the time they appear to be regulatory and having to do a bit with what Stephen presented this morning, affecting the expression of a gene. So this was the first loci we identified in a consortium. Celia Greenwood who's here at McGill, did the genetic association at first GWAS for colon cancer. This was at the time when we started 1,000 cases, 1,000 controls. We thought 1,000 was large. We know now that's not true in this area. But in Ontario, we found a locus which had higher risk in cases, an allele which is relatively common, but we needed to actually validate in about 10,000 cases and controls from different cohorts before we got genome-wide significance. And it's just to give about a 17% increase in risk if you have one risk allele and 34% if you have two. Not very informative for the clinic. When we started, had 10 loci which had been validated in over 10,000 subjects were past genome-wide significance. We started just counting the number of alleles a person had. So if there's 10 markers, there's possibility of inheriting 0, 1, or 2 risk alleles at every locus. So a genome could have 0 to 20 alleles. We never see those extremes. But the most common rate we see is 9 risk alleles for colon cancer in the population. So we set that odds-odd ratio of 1. And you can see that if you have 13 or 14 alleles, your risk is about a 2-fold increase. And if you have 4 or 5 risk alleles, so very little, compared to only about 1% of the population, your risk is around 0.4. So we're starting to discriminate high-risk and low-risk groups. When we started looking at 40,000 individuals, but looking at other risk factors like age, like gender, we see a lot more. It becomes even more informative. And family history, because family history is not all accounted by 10 or 20 or 30 GWAS SNPs. So in this And this example, for example, these are males, which would have 14 risk alleles. These are for the first 10 markers, 14 risk alleles and a family history. So their risk, if they're 50 of getting colon cancer in the next decade, is 10%. If they were 60, it's about 40%. So now we're starting to see some discrimination of groups which are at high risk for individuals. Even 12, but this is only about 1% of human population, but having 12 or 13 alleles plus a family history increases your risk about 15 or 20% of having colon cancer. So we're at the point right now, all these data are retrospective for these large case control studies we've collected over the years, but we suspect that this is probably groups which would have colon cancer screening starting earlier or more frequently, and this is where some of the screening programs, research studies are happening right now to try to link and identify high-risk groups or low-risk groups and try to modulate the frequency of screening so again we can pick cancers earlier and of course save lives through that process. It's not really a topic of my talk today, but I just wanted to continue but because we heard about genome-wide associations and exposome this morning, but we just had this paper as our consortium of colon cancer on risk for aspirin. So we know aspirin protects against colon cancers, about 30% protection. So it's actually a big effect, a way to prevent colon cancer. And this paper that just came out in JAMA said that 95% of us actually have an allele which give even a higher protection than 30% if you take aspirin every day. But 4 or 5% of us have alleles that actually, if you take aspirin, your risk is higher. So it's an editorial in JAMA actually saying how things can be complicated. And this gene that was identified as a gene has to do with the metabolism of prostaglandins, and aspirin, of course, is important in those pathways. So even a risk allele could be protective, taking aspirin could be protective at certain genotype and risk at other genotypes. So how are family physicians going to use that information? I'm not so sure. But here I said I'd talk about early cancer, so I want to talk polyps. We looked here just to see, are the risk alleles that we see for colon cancer the same as for polyps? And in fact, if we look at individuals with polyps which don't have a colon cancer, we actually see that they have about the same risk allele score as people with colon cancer. So there certainly is a very similarity, a big similarity in terms of risk for polyps as there is for colon cancer. This locus, which I'm going to show you, is locus on chromosome 11, Q23, is the most strongly associated locus for having polyps, for adenomatosis polyposis. So this is one of the many loci that have been identified by GWAS studies, but it's associated with an early cancer called polyps. And it has 3 genes. So this region I'm talking about, I'm going to show on the next slide. Basically, there's 3 genes. There's a region of about 12 kb, which are high linkage disequilibrium, and there's 3 genes that overlap, and as you'll see in a minute, all the 3 genes are co-regulated by this region. And the genes, they were called opening reading frames, so they're predicted by the Genome Project, but they hadn't been studied before. COLCA1, we changed the name from ORF92 to COLCA1, colon cancer associated 1. COLCA2 is colon cancer associated 2. And this other 3rd gene, I wasn't really thinking it was a good candidate for different reasons, but now we think it is. And I'm going to show you why in a minute. So we started first doing type of expression studies as were mentioned by Stephen this morning, looking for AQTL. And for all 3 genes, you can actually see the genotype correlates with different levels of expression. So for ORF53, COLCA1, and COLCA2, having the protective genotype, you express higher levels for all 3 genes. So the genes are basically co-regulated by a common regulatory element. The genetic variation that we have actually makes that all 3 genes are co-expressed at higher levels or lower levels at the same time when we look at colon cancer tissues. But one other thing I can tell you, they're not expressed in all the same cells. So it's not completely co-regulated, but that's another story. And I just thought I would tell you this is a story evolving, but one of the first things we notice is that this locus, when you look at colon tissue, even normal tissue, the histology is different when you compare people at risk and people not at risk. That will be one thing. I'm going to show there's much more lymphocytic infiltration in people which do not have polyps. I'll show a little bit of characterization of genes, and I'll show COCA1 just to give you an example that not all the genes are important in cancer cells. Some appear to be important in immune cells. Again, immunity is important to actually prevent colon cancer. And I'm not going to show results, but more recently I dismissed the third gene because we didn't see it initially expressed much in colon cancer. But Philip Gros at McGill actually had published a paper on a mouse locus for colon cancer, and through different ways we were able to say that this gene is in the region of interest for his project, and he sent us tissues, and actually we saw the same level of higher expression correlating with protection. So again, 3 different genes. Each of them have a potential associated with colon cancer risk. Each of them are higher expression, and the molecular biology we do of the 3 different genes all link them to colon cancer pathways. To able to distinguish whether it's one or all 3 genes, we have actually mouse constructs which are underway. So this slide is just to show you that what I mentioned about lymphocytic infiltration. Our pathologist, when we were giving him different tissues from people which were protected or at higher risk, noticed there were more lymphocytes. So we're using a red marker here for lymphocytic infiltration, and you can see on the top is histology of colon mucosa, far from the tumor, close to the tumor, or within the tumor. You see a lot more red on top than at the bottom, and that's someone which has a higher risk, the protective genotype on top, and lower risk genotype at the bottom. Just to make sure it's not just a figment of our association. We might just pick parts of the tumor that we think are important. When you look at large numbers of tumors, you actually see this correlation. So protection correlates with higher risk, higher degree of lymphocytes. And that's also something that's been associated. Generally, people with colon cancer have more lymphocytes, actually survive longer. So it actually correlates with some other epidemiological data. When we started studying this first protein, we got an antibodies. We actually discovered it wasn't in the tumor cell, it was in immune cells in the gut. Half of the eosinophils, that's a type of white blood cell in our body, are in the gut. They're very important. There's a lot of bacteria in the gut, of course, so we have a very sophisticated immune system in the gut. And this red here right now is in the granules of eosinophils, type of white blood cells. We see the red is actually COCA1, but we see that in mast cells, macrophages, neutrophils. Here, something else that was kind of novel, and I didn't know because I am an immunologist, but when I went through medical school, I learned that eosinophils just liberated their toxins close to allergies or close to parasites. But we actually see in these slides that this doesn't work for, okay, that we see these granular structures, and these are different immune cells. We can actually see that the granular structures are actually excreted by the eosinophils and actually migrate through the immune tissues. So we have cell-free granules which contain our proteins, which are actually diffusing in the intestinal tissues, and it's probably a way to get the effector molecules very close to the pathogen, or in some cases to tumor cells. This is again COCA1, just more to prove, and pathologists always have a trouble understanding that we're studying a colon cancer tissue, but colon cancer gene, but it's not in the colon cancer cell. It's in the microenvironment, and you can see more red on the top and on the bottom. Again, there's association in different tissues across hundreds of samples. So this project is not finished, but I just wanted to say that it's one thing to have a hit, to actually have an association, but it's a lot more work to go and study the biology. And here we're studying the biology, finding new immune mechanisms important in prevention of colon cancer, prevention of polyps. Where this may lead to, of course, new ways of thinking of chemoprevention, new ways of combining that with aspirin. But at this point, this is probably where the state of the GWAS is actually needs to really think through. We've identified thousands of loci, and if we want to really know what the biology means, we're going to need to study every one of those loci one by one, as we've done here. Now I'm going to talk about the tumor genome. So when we went to medical school, we often learned, and still true, we learned it in high school, and that we have 2 chromosome 1, 2 chromosome 2s, and so on. But when you look under the microscope, we've known for a long time, there's a lot of chromosome abnormalities in cancer. So this cell here has 6 chromosome 2s, has rearrangements, has deletions of chromosomes. Now when we sequence tumor genomes, we find typical cancer genome has on average 10,000 mutations that are not found in the inherited genome, so a large number. If you're a smoker and you have cancer, your tumor probably has 100,000 to 1 million mutations. Actually, it's about one mutation per 15 cigarettes smoked. So if you actually divide the number of cigarettes per lifetime, so there's a lot of mutations in cancer, and if you're exposed to carcinogens, you have more. But we see lots of abnormality. We didn't suspect that high rate. Now, most mutations are neutral. They don't have any effect on the biology, but if they fall on a specific gene or specific site, they actually can drive the cancer process. We call them drivers. So the ICGC was started, International Cancer Genome Consortium, just before we sequenced the first cancer genome, because we knew that if it cost us billions of dollars to sequence the first genome, but it was getting cheaper and cheaper to sequence genomes. We knew there would be abnormalities in cancer, and we thought it would be important to catalog all the cancer mutations. So we did start a big project to resequence 25,000 cancer genomes. The project involves sites across the world. We've, again, 85 projects, most of them trying to sequence 500 tumors of a given type. We compare, for example, liver cancer to viruses in Japan and liver cancer to alcohol in France, and different exposures, different disease. We've had 2 projects. We've discovered a novel carcinogen because when you look at the DNA patterns, depending on what carcinogen, you can see different types of mutations. So I can look at DNA and say, this person's a smoker. So it's possible to look at these people had different carcinogens, and it's a project that came out of McGill University in France. One of them is aristolochic acid. It's a food extract, but actually has a carcinogen and causes more kidney cancer in the Roma, and also more bowel cancer in Thailand. It was identified by a sequencing project. So we are learning more about the genomic environment, not just the genomics. So these are the data sets which are available. These are the growth in the cancer genomes, about 12,000 cancer genomes on the OICR website. And I wanted to show 2 examples of studying early cancers once you have access to these data. The first one is a project in leukemia. Now, one of the ICGC project was made in the US. It's a leukemia project, AML, acute myeloid leukemia. And I want to just point out that these catalogs of mutations that came out from these groups showed that there were a lot of genes. Some genes are more commonly mutated, like FLT3 or NPN1. But some genes, I'm going to discuss them a bit later, genes that are important for the epigenome. They control methylation. They control chromatin structures, which are important in gene regulation. And so DNMT3A and TET2 are examples of epigenetic genes which were found to be mutated in leukemia. We don't know the whole process of going from a pre-leukemia to leukemia. We know a few syndromes, but most of it we don't know. But we do know that when you look at a final leukemia, you actually find many mutations, as we do for many different cancers. But there's a question, is there an order at what mutations come first? Would it be possible to identify people with a pre-neoplastic, almost the equivalent of a polyp, of an early disease, before they actually have acute disease? So in the project that we have, it's funded by Genome Canada, funded by CIHR, it's called the Cancer Stem Cell Consortia. John Dick is one of the scientists at OICR, has been studying cancer stem cells. We talk about stem cells for the brain, stem cells, they give rise to the bone marrow, but we also have in tumors, we have cells which have the potential to, when isolated, to form a whole new tumor. Few cancer cells have that potential, actually. So we're trying to understand what are the stemness properties of these cells, which are important in metastases, in cancer, in resistance to drugs, and so on. We used the TCGA project to make a panel of all the commonly mutated genes in leukemias, and we decided to sequence the leukemia of patients. And also, we needed a normal tissue. Now, usually in most cancers, the normal tissue is blood, but when you have a blood leukemia, you have a problem of trying to, we want to have the normal DNA. So since these people have AML, we were able to isolate pure T cells and use that as a source of normal DNA. And this is a large panel, but I just want to show on the top are the names of the genes which are part of the panel, which we're resequencing. And going down, we have different patients. And for each of them, we sequenced the AML, the leukemia, and the T cells. And what's interesting here, it's hard to show them with my thing, is that we started seeing that although NPN1, which is over here, we see a lot of mutations, but only in the AML cells, not in the T cells, as we expected of a tumor mutation. We started seeing that for DNMT3A, we had 4 patients. We found the mutation not only in the leukemic cells, but also in the T cell. That doesn't make sense. It's supposed to be the normal DNA of an individual, of a cell. These are clearly been purified not to have leukemic cells, yet they're seeing a tumor mutation. And so then we were able to go in the fridge, look at more samples which had both these mutations, NPN1, which I'm going to show you is a late mutation, and DNMT3A, which is an early mutation. And we were able to see that we could, DNMT3A and a few other genes like TET2, we could see them not in leukemic cells, but also T cells. Now we know our bone marrow gives rise to all types of blood cells, T cells, B cells, NK cells, or red blood cells, or platelets. So we started wondering, well, maybe that's where the mutation comes from, an early mutation in the bone marrow that actually gives rise to a normal blood system, but in addition gives a growth advantage to some cells to form tumors. So John's been studying the human lineages for years, and I've created different ways of isolating every cell types and all progenitor cell types and stem cells for the blood system for years. And we were able to get this from different patients with leukemia. And what we do here, basically just want to show, is that when we look at the acute blast is the acute leukemia. When we look at these 2 mutations, if it's black, we're seeing both NPN1 and DNMT3 mutation, both the late and the early. But when we're isolating the fraction of cells which are supposed to be stem cells, we're only seeing the DNMT3A. So again, suggesting that we have an early mutation that happens first in this set of genes is found in the stem cells. You produce a normal bloodstream, and over time, some cells that are important in the myeloid lineage have additional mutations and give rise to leukemia. We're able to, here I'm showing the size of the circle is the frequency of that mutation when you look at all the cells in the body that we're able to isolate, B cells or T cells. So when a patient had a leukemia, in this case, we could see that stem cells, B cells, and this is the blast fraction, had a lot of DNMT3A mutation. You treated the patient, all the NPN1 mutations disappeared, so the leukemia is gone, but we're still left with many cell types or actually still have a tumor mutation in them, which are a signature of that precursor mutation. And as this patient, this is now 3 years later, still doesn't have the leukemia, but we continue to see that that fraction of clones is now the predominant source of the bloodstream. So likely we are having an expanding reservoir of pre-leukemic cells, and likely it's going to be the source for the next leukemia of this patient. So what have we learned about early cancers? We've learned that most cases, of course, what we knew already, we have stem cells in our bloodstream that give rise to a whole blood system. But in some cases, you can have an early event. And the early event can still produce cells which can give rise to lymphocytes and platelets and red blood cells. But overall, over time, these cells have a growth advantage, become a reservoir for additional mutations and growth, and is actually the source for the tumor happening, and that even when you treat the leukemia, these cells stay behind and are the source for the second mutation. Come back to that later. Third quick story, prostate cancer. Prostate cancer is very common in men, and over time, it's the #1 cause of death, very common. And actually, most men have prostate cancer at autopsy, even if they die for cardiovascular disease or others. So it's a very common disease. But when you distinguish prostate cancer, we know that more than 95% will still be alive 5 to 10 years later. So it's actually very difficult to make a clinical decision whether you're going to do a surgery, give chemotherapy, or just watch while waiting. So over time, we've used clinical criteria to actually make a distinguish whether we would use like pathology, we use PSA, we use other clinical information to decide whether patients are into 3 different groups. The top group is the low-risk group, and low-risk group have only active surveillance. They don't have surgery or chemo. They would just follow them up over time to see, and maybe they're just to see whether the small tumor will actually grow or not. There's groups with more aggressive disease pathology, for example, especially Gleason 8. These are pathology scores. Actually, we'll have the prostate removed, and have adjuvant therapy or radiation therapy. And there's an intermediate group which actually has a surgery without the additional radio or chemotherapy called the intermediate risk. And what we're asking in this project here is whether people with intermediate risk might be two different groups. A group which is less aggressive disease and should have only active surveillance, and a group, or maybe groups that should have more aggressive therapy. So in our ICGC project, we decided to sequence 500 tumors of patients with intermediate risk. So we had a large cohort of 2,000 men, which we had biopsies that had been collected 5 to 10 years earlier. We were able to select the intermediate risk subgroups. We have outcome information. We sequenced their genomes. Because this is probably one of the most difficult decisions for a clinician and a patient. So I'm going to talk about a radiation therapy cohort. It doesn't really matter, but we did use different cohorts to validate, both from Canada and UK and US, to validate some later results. I'm only going to talk about one type of genomic abnormality called copy number variation. That's where the number of copies of a gene, which we should have 2, could go, if there's a deletion, you have 1 or 0, or if it's amplified, you can have 3, 4, 5, 6, 7. So I'm looking for changes in the number of copies of a gene. And so this is what I've just mentioned here. When we look at copy number changes across the genome, we actually see different things. So you see some tumors which have very few changes, so you can mostly white. You have some places where you have deletions, which are in blue, and some places you have in red, which is an amplified. And if you compare the genomes compared to the normal this is different ways of looking at the clinical correlates. There's not a huge, it's hard to see patterns that actually correlate between the clinical criteria which are used and the genomic criteria at this point. When you use machine technologies or algorithms to say, well, are there patterns within those tumors, it actually breaks down the patterns, the types of tumors into 4 different groups. The major one, which is down here, there's people which have almost no changes, mostly 2 chromosomes across the tumors, so very little chromosomal changes. But there's 3 other groups. It's easier to see in this slide. The group of cluster 1, basically you see a lot of amplifications and gains. So this in chromosome 8, for example, a lot of amplifications, chromosome 7 here. Cluster 2 is mostly deletions. Cluster 3 is mostly deletions. Cluster 2 is a mix. And cluster 4 is about 50% of the tumors are actually mostly normal. When you look at the outcome of these different groups, you actually see that depending on your genomic abnormalities, you have different outcomes. And here, for example, the biggest group with almost no abnormality do much better over time, over 5 years, than the group which are at the bottom here. So we decided to do a test, just the percentage of genomic abnormality in a tumor. Can we use that as a predictor of long-term outcome? And the answer is yes. Is that if you just take the percentage, less than 7% genomic abnormality, people do quite well in blue, and if you have more than 7%, the risk of recurrence, so this is biochemical recurrence, is actually much worse. So number of genomic abnormality is a predictor of response. And here, this is a more sophisticated way of looking at 100 places in the genome, which would be the best predictors, and again, you can distinguish high risk or low risk. And compared to all the other biomarkers that have been published so far, it's the top one. It's the best biomarker. It's the best predictor of long-term outcome. So if you come back to my last question, I know I'm finished. I asked you a question: could we actually distinguish this group here? Say maybe we should have a group which have less aggressive disease, and maybe there's a group which have more aggressive disease. In fact, our results so far, this was published in Lancet Oncology, is that about 50% of patients in this group appear to actually have as low risk as what we would call it traditionally a minimal risk, so probably should not have chemotherapy or adjuvant therapy. Sorry. And the other 50%, we're a bit less sure how to do the interpretation, appear to have more aggressive disease. And so we are starting to use genomics as a way of early cancers to provide some information that might be clinically important in terms of disease management. So I'm going to stop here. I just want to point out that, again, that genome variation, the biology, tumor microenvironment, all of these things are integrated. It's not metabolomics versus genomics or epigenetics versus immunology. All this information is really important to try to get the big picture and put this information, which is important for the patient in terms of outcomes. Again, many different people in my lab to thank and many people in the prostate cancer project. Thank you.